Thank you so much, Uma, for your interesting presentation. But this wasn't the only way proteins can help with sustainability. Adrian is using them in a study to create biophotovoltaics, which use proteins to convert sun power to electricity. Thank you.
Great. Thank you very much, Sana, and also thank you very much to the organizers for giving me a chance to present my research here. So my name is Adrian Bunzel. I'm a junior group leader at ETH Zurich, and I'm interested in creating new proteins for sustainable applications. And so specifically, I'm interested in using computational design to create new proteins that can harvest sunlight and then use the energy of the harvested light to produce electricity. So in a nutshell, I want to make a biological solar cell. Now, obviously, my system is not going to look like this. This is just an illustration. But using biology for solar-driven applications is actually not such a far-fetched idea. Because if you think about this, nature has been doing this for billions of years in a process called photosynthesis. And photosynthesis is inherently a protein-driven process. So on the right over here, I'm showing you a photosynthetic protein. The protein is depicted in gray. And I'm showing a couple of small molecule cofactors that are bound to the protein. And it's really these cofactors that drive the photosynthesis. So if we have a closer look at the cofactors, you can see that they're all beautifully aligned by the protein to harvest sunlight and then get the photosynthesis going. So as soon as light or a photon is absorbed at this green cofactor down here, an excited electron is produced, a high-energy electron that is transported through this electron transport chain to kickstart photosynthesis and get the metabolism going. Now, because the protein scaffold itself really controls all steps along this electron transfer chain, natural photosynthesis actually has almost perfect quantum yields. That means that every photon that hits this cofactor over here is actually converted into a high-energy electron. Unfortunately, though, natural proteins are fairly complex, not very stable, and they also have been optimized by nature for their native context. So they tend to not to work so well anymore once you take them out of the cell into an abiological context in a biohybrid device. So what I want to do in my research, I want to create new photocatalytic proteins that I can then apply in solar-driven devices. And before I tell you about the specific proteins that I've been making in my research, I want to very briefly comment on the process of protein design. Because designing new proteins is not simple at all. If you think about it, even a very, very small protein can have an astronomically large number of different sequences. To give an example, if you have a 100 residue protein, and in this protein, at every position, every residue can be 1 out of 20 amino acids. This gives you 20 to the power of 100 possible sequences. And this is a number that is much, much larger than all of the stars in the universe. So we need to come up with smart ways how to screen this sequence diversity and identify sequences, identify proteins that will actually behave the way that we want to do. And in the past decades, computational chemists have developed a lot of really powerful approaches to do so. And in the simplest one, we typically start by modeling an existing protein, and then we introduce mutations, one at a time, one at a time, one at a time, until we change the properties of the protein so that it really does what we want to do. And although this sounds a little bit tedious, this has been an incredibly powerful approach which people have been using for decades now, very, very successfully to create new proteins and even new enzymes. I'd like to very briefly comment on the fact that artificial intelligence has really revolutionized how we do protein engineering and protein designs nowadays in the lab. Some of you might have heard of AlphaFold. There's something missing over here. Some of you might have heard of AlphaFold for structure prediction, but we can also use AI methods for protein design. So nowadays, it's really possible to draw up the structure of a protein in the computer, ask the computer to give you a sequence, and you test the sequence in the lab, and you have very, very high success chances that you actually end up with a protein that looks exactly the way how your AI algorithm has predicted the structure. Now, coming back to my original design idea, so just to remind you, we are working with these natural photoenzymes. They are photoactive, but they're fairly complex and unstable. So the very, very simple idea of my research is that we want to start with an enzyme that already exists, or with a protein that already exists, that is very simple, much, much smaller, very stable, very well characterized, but doesn't have any photoactivity. And then we use computational design methodologies in order to introduce photoactivity. So the idea is very simple. We want to design a new binding site for a small molecule photosensitizer for another cofactor that will absorb the light and then kickstart electron transfer. And this is exactly what we did. To cut a very, very long story short, I'm showing you the structure of one of the proteins that we actually have designed down here. So we added this green cofactor, which is really geared at absorbing light and then transferring an electron to this orange cofactor over here. Now, we tested this protein. It produced really well in E. coli. We could see binding of this green photosensitizer. And now the big question was, does it have any photocatalytic activity? And to test this, we assembled a model biological solar cell where we took our protein, just sandwiched it between two electrodes, and then we're shining light on it. And what we're doing here is we're measuring the current that is formed upon excitation with an LED. And if we switch the LED on, we see a current. If we switch the LED off, we lose the current. And this is really, really encouraging, and we were absolutely thrilled to see this. Now, admittedly, this is not perfect yet, so we probably need to improve this by roughly 50 to 100 times to really become competitive with state-of-the-art photovoltaics. But I'm actually mildly optimistic that this is going to work, given the huge advancements in both computational and experimental protein engineering, and also given the fact that this is a fairly naive scaffold where we didn't yet do much of engineering. To conclude my talk, in my talk today, I've pretty much focused on electricity. But I believe that protein engineering can really be valuable in a lot of other fields to give rise to a more sustainable future. In terms of energy, we're currently exploring the photosensitization of other enzymes that exist in nature that already have some catalytic activity, and then we want to use computation to imprint them with photocatalytic function. So the idea here would be that we create solar-driven systems to create biofuels, to create fertilizers, or to capture CO2. And with that, I'm at the end of my talk. I'd like to thank all the group members that have helped in this research, and I would like to thank you very much for your attention.